introduction to the process of respiration. So, respiration is the release of chemical potential energy from food. The energy released is used to form ATP from ADP and inorganic phosphate. And we can usually represent that reaction like this. So ADP is phosphorylated by inorganic phosphate to form ATP. Okay, so this here is inorganic phosphate. What that means is that it is usually phosphate ions that are floating around the cell cytoplasm or in the nucleus, wherever it's needed. Okay, this process we call a phosphorylation. And we always say that ADP is phosphorylated to ATP, or ADP is combined with inorganic phosphate to form ATP. We never say ATP is formed. You must include those two reagents as well. Okay? There are two types of respiration. Aerobic, which uses oxygen, and anaerobic, which does not. The chemical equation for aerobic respiration is the reverse of photosynthesis. So basically, we get hexose, so glucose, that is combined with oxygen to form water, carbon dioxide, and then energy is released. Now that looks a simple one-step process, but that process is divided up into different stages, each of which has got many chemical reactions. A lot of this is going to be similar to what you saw in photosynthesis. But in general, what happens in aerobic respiration, and we'll talk about anaerobic respiration a little bit later, but what is happening in aerobic respiration is the breakdown of glucose molecules or hexose molecules. So for example, hexose is C6H12O6. And respiration involves the process of removing hydrogens from glucose molecules. So if you keep removing hydrogens, eventually you're going to end up with carbon dioxide, and you'll also end up with some water. Now those hydrogen ions, not drawn here, but there would be ions, also will have some electrons associated with them. And it's these that then go to other processes to release energy and that energy is going to be used to combine ADP and inorganic phosphate to make ATP, which is what we're after. So the important thing about respiration is it is the slow release of hydrogen ions and electrons. The energy is passed to form ATP from ADP and inorganic phosphate. This bit is very, very similar to the light-dependent stage of photosynthesis, where you had electrons that contain lots of energy moving between carriers in a membrane. This bit is very similar. This bit is identical to chemiosmosis that is taking place in membranes. Same process as in photosynthesis. So, there are a number of stages involved in respiration, and here are the different stages. And in fact, aerobic and anaerobic respiration start at the same point. And that same point is hexose or glucose. So we've got C6H12O6. That is our starting point. The first stage is a process you might have heard of before called glycolysis. And that takes place in the cytoplasm of cells. That process involves a number of stages. It involves making ATP. It also involves removing hydrogen. Now, for this overview, I'm not going to be interested in the finer details of the chemical processes that are taking place. We're just seeing the overall processes and where molecules are going. Okay, we're going to be looking in subsequent lessons at each of the stages in a little bit more detail. So that first stage, glycolysis, we get glucose or hexose, let's call it hexose, and it loses hydrogen. It forms ATP from ADP. It gives it a phosphate group, inorganic phosphate group, and we get formed at the end a substance called pyruvate. Now hexose, of course, is a 6-carbon compound. Pyruvate is a 3-carbon compound. You do need to know the structure of hexose, glucose. You do not need to know the structure of pyruvate. So that process takes place in the cytoplasm. Pyruvate has 2 possible destinations. If there's no oxygen present, then that will go to anaerobic respiration. And I'm not going to talk any more about that in this video. However, pyruvate does then go to another stage, and that other stage involves pyruvate being converted to a substance that contains acetyl groups. And that process is called the link reaction. And what happens in the link reaction is that we get carbon dioxide released, and we also get some hydrogen released. Now, I've written H2, it looks like hydrogen gas. Of course, it's not hydrogen gas, but I'll talk more about that in a moment. The link reaction takes place in the matrix of mitochondria. So this is taking place in the cytoplasm, the pyruvate that is made by glycolysis moves in to the mitochondria to the middle, which is called the matrix. And we get acetyl groups formed from that. Those acetyl groups then enter another process. And this process is a cycle. And this is called Krebs Cycle. It's a series of enzyme catalyzed reactions, as all these are, and it's a cycle. So it's continually going round and round and being fueled, as it were, by acetyl groups coming in. Now, a number of things happen during this. We get some carbon dioxide released. We get some more hydrogen, quite a bit of hydrogen released actually, in different places. We even get a little bit of ATP made. So that too, it's taking place in the matrix of mitochondria. The last stage involves using the hydrogen that has been removed from the hexose. So we've got 12 hydrogen here coming in, and if you look, we've got 12 hydrogen altogether coming off. You don't need to know the great detail about that. Now, the important thing here is that hexose contains a lot of chemical potential energy. And that chemical potential energy is really associated with the hydrogen. So by the hydrogen slowly being removed from the glucose, we are slowly removing energy, and we're able to use that energy elsewhere. So what we have is all these hydrogens. And those hydrogens have to be moved to somewhere where they are able to have their energy released and put to some use, i.e. to make ATP. And the place that happens is in the inner mitochondrial membrane. So let me just do a little reminder of the structure of mitochondria. Mitochondria are made of two membranes, an outer membrane, which is pretty boring, and then an inner membrane, which has got lots of folds on it. And these folds are called cristae. And they project into a semi-fluid center. And so this semi-fluid center, that is the matrix. So that is where the link reaction and Krebs cycle is taking place. So the cytoplasm is outside here. Glycolysis is taking place. We get pyruvate made. That pyruvate moves into the matrix of the mitochondria. Throughout these locations, we have hydrogen being released. And they are moved to the inner mitochondrial membrane. And in the inner mitochondrial membrane, or on it, are little structures, which I'll talk about later. And on the same scale of this diagram, they would be so small you wouldn't be able to see them. They would be less than a dot. These structures and the membrane carry out another process. And what basically happens is that the hydrogens are carried to the inner mitochondrial membrane, and they are all carried to the same, more or less the same place, by a group of molecules called coenzymes. If you remember from photosynthesis, coenzymes are molecules that are able to pick up small atoms, small chemical groups, and move them from one place to another. So in photosynthesis, the coenzyme was NADP. There are two coenzymes that are involved here. One is called NAD, no P, and FAD. And as I say, we'll talk more about them a little bit later. And they go to the inner mitochondrial membrane, like we have here. So it moves to another process which takes place on the inner mitochondrial membrane. And basically what happens is that we've got lots of hydrogen. That hydrogen is split into hydrogen ions and electrons. And then those electrons have got lots of energy in them, chemical potential energy. And they are passed between a series of carriers until they have less energy. And as they are passed between the series of carriers, energy is released. And that energy is used by the process of chemiosmosis to combine ADP and inorganic phosphate to make ATP. So what we have at the end is some electrons and some hydrogen. We need to get rid of those. So what do? Let's get an oxygen molecule. Let's get an oxygen molecule even. And combine it with them. So we're going to get half an oxygen molecule, which is an oxygen atom, and we'll get water formed. And that is the only reason why we need oxygen, to finish off the whole of the process. So that process is called oxidative phosphorylation. And this process, which is a part of it really, is chemiosmosis, which is exactly the same as what we discussed in photosynthesis. The first process we're going to look at in a bit more detail is glycolysis. You need to know some of the molecules that are involved. You don't need to know any of the structures of these molecules, apart from the starting molecule, which of course is glucose or hexose. So we start off in the cytoplasm with hexose. And hexose, which is, if you remember, C6H12O6, could be glucose. It could be a number of different 6 carbon atoms. There's a number of them that it could be. So we generally use the term hexose to show that it isn't just glucose that could enter glycolysis. The first thing that happens is a little bit strange, because remember, the function, the purpose of respiration is to release energy to make ATP, so that energy can be used for other processes. But the first stage in glycolysis, in respiration, is actually using ATP. And what happens is that ATP comes in and gives phosphate groups to the hexose molecules, which seems a bit strange, really, but bear with it. We end up with a substance called hexose phosphate. Sometimes we call it hexose bisphosphate. It's got 2 phosphate groups in it. So for that to happen, we need to have 2 ATPs coming in and we have 2 ADPs coming out. Those hexose phosphates then split into triose phosphates. And those triose phosphates then undergo a series of chemical reactions forming pyruvate. Now, as I said, you don't need to know the detail of those chemical reactions, but I'll go through it just to make sure. The first thing that happens is that some inorganic phosphate is added. Don't worry about where that comes from. It could come from the diet, it could be absorbed through the roots of plants if we're talking about respiration in plants. The next stage is the removal of hydrogen from the triose phosphate molecules. And those hydrogen molecules are added to the coenzyme NAD. And when NAD picks up the hydrogen, it forms reduced NAD. After that, we get some ATP made. And the ADP picks up phosphate groups that have been added to the hexose to form ATP. And we end up with pyruvate. Now, the only thing you need to be aware of are the names of the reactions that are taking place and how many molecules are involved. So here we've got 1 hexose molecule. 2 phosphate groups are added to that hexose molecule. So we call that reaction a phosphorylation. Hexose is being phosphorylated. Hexose phosphate is produced. And I'm going to put here the number of carbon atoms that are in that molecule. Still 6. We know there's 6 here and there's 6 there. What happens is that this 6 carbon sugar with phosphate groups is broken down into 3 carbon sugars with a phosphate group. That is called a lysis reaction, breaking down. But because of that, we have 1 6 carbon and we get 3 carbon compounds made, then we have 2 of those produced. So from this point onwards, everything is doubled. So what we have is 2 triose phosphate molecules. Each of those is a 3 carbon sugar. So if I drew a carbon atom as a circle, each of those would be 3 carbon atoms, and each of them has got a phosphate group attached. What happens now is that for each of them, another phosphate group is added, the inorganic phosphate. So we have to say 2 inorganic phosphates come in. And that, therefore, here would also be a phosphorylation. Then each of those molecules, so we would now have that's what we had, and we've added an extra phosphate to each of them. Don't worry how that happens, it just does. Then each of those triose phosphate molecules lose some hydrogen to NAD, and because we've got two of those, then two NAD come in and we get two reduced NAD coming out. That is an oxidation. Because we are losing hydrogen. We can also call it a dehydrogenation. So finally, we're left with molecules which have got 4 phosphate groups on them. And each of those phosphate groups can be removed and added to ADP. So we get 4 ADP coming in, and each of those takes away a phosphate group. So we get 4 ATP formed at the end. And then finally, because we started off with 2 triose phosphates, we end up with 2 pyruvate molecules. And if you look, there's no loss of carbon. So each pyruvate molecule is made of 3 carbon atoms. That process is a dephosphorylation. Now that's a dephosphorylation of the molecules, triose phosphate or whatever is remains of them, because other things have been going on before here. And it's an example of substrate level phosphorylation. Now that might not make much sense at the minute, but it will once we finish, and I'll come back to that right at the end. But what is important here is that we are making ATP without any oxygen being added to this whole process, and that's quite important. So before we leave glycolysis, let's just think about the important points. We have the phosphorylation of hexose or glucose to hexose or glucose phosphate using ATP. So we're using up a bit of ATP here. We get triose phosphate molecules formed, and then those are oxidized to form pyruvate with ATP being produced and reduced NAD being produced. This is very important. This is going to be used later on to make lots and lots and lots of ATP. But the process of glycolysis allows a little bit of ATP to be formed here, which is always useful. Now, what is very useful is to think about how much ATP is being made overall in the process. Okay, so we can talk about the net ATP production, and net means overall, which is basically the gross AT production, which is how much is being made, minus ATP that's being used up. So let's have a little look and see if we can work this out. how many ATP molecules are being produced in total. So we've got some being used up there, but here we've got 4 at the bottom. How many ATP molecules are being used up? 2. Which means that the net ATP production is 2. The overall production of ATP in glycolysis from one hexose or glucose molecule is 2. That is the process of glycolysis. And at the bottom, we end up with pyruvate, which could go to anaerobic respiration, and we'll talk about that a lot later, or can go on to the next stage, which is the link reaction, and that's for next time.